脑人言，
各位朋友，大家好啊！今天非常荣幸呢，我能邀请到我的同事于工程博士来介绍他最近发表在 Neureport 上的文章。于博士12年呢本科毕业于华中农业大学，19年在中科院神经所拿到博士学位，随后他来到了 NIH 眼科研究所的 Richard Classilis 实验室做博士后研究。他的研究内容呢，主要是关于面孔信息的神经环路编码机制。今天呢，介绍的是他最近发表的在 SC 上面的一部分的研究成果。然后我很久以前我就听过他的报告了，我觉得非常的精彩，所以我看到他的文章发表以后呢，我就赶紧把他邀请来，请他来介绍一下这样的工作。那接下来时间我们就交给于博士了，郭生你开始吧。
好的好的啊，首先非常感谢老人言还有唐华老师的邀请，今天非常开心就给大家就是介绍一下我们最近的工作，然后就是我这个接下来讲的过程中，如果大家有什么问题就可以随时打断我，然后虽然我这个讲座是英文，但是就大家问问题中英文都没有什么问题。OK，okay, now let's start. Hello everyone. Today, I'm Gongchen Yu from Laboratory of Sensorimotor Research at National Eye Institute. Today, I'm very happy to share with you our recent findings about short latency preference for faces in the primate superior colliculus. As primates, we always like watching faces, and we are actually very good at processing faces. So far, the best understood neural mechanism about face processing has been largely focused on the cerebral cortex. So here is a lateral view of a monkey's brain. So if you show the monkey face and non-face stimuli, you will see several brain areas in the temporal cortex will be selectively activated to face, but not to non-faces. So these are the well-known temporal cortical face patch system. So it has been well established that this temporal cortical face patches, they play a crucial role in recognizing a face, such as face identity and the facial expression recognition. But however, the human subjects, they can already selectively mix the cars towards faces over non-faces at a speed that is even faster than the cortical face patch and process them. And also a very recent work, they found in activation of the cortical face patches in monkeys actually doesn't impair their ability to find and look at faces in the visual periphery. And it's more shocking that in the primates newborns who have yet to develop cortical face patches, they already preferentially look at faces, for example, their parents' faces. And more interestingly, for these babies, they need to keep looking at face. They need to keep having exposure to faces for another 6 months to finally develop the cortical face patches. So all these findings just suggest the existence of other mechanisms that are important for rapidly detecting and orienting towards faces. So today, I'm not going to, that's why today I'm not going to talk about face processing in the cerebral cortex. I'm going to share with you our findings about face processing in a subcortical structure called superior colliculus. So here, let me give you a brief introduction to this subcortical structure. So first, the superior colliculus is an evolutionarily conserved structure on the roof of the brainstem. So it has been best known for its role in controlling where we look, which is eye movements. But however, the superior colliculus is not only a motor structure, it is also very important for visual processing and forms the salience map of the visual space. For example, this left superior colliculus, they cover the right side of our visual space. And the visually responsive neurons in the superior colliculus, they all have retinotopic receptive field. And these neurons, they really care about the visual stimuli that are physically salient due to low-level features such as a higher contrast and luminance. And there are some recent findings. They also find as the superior colliculus, they may play a role in processing face-like stimuli, which are pretty interesting. But however, it is still unclear to what extent these recent findings, they depend on the visual salience of the stimuli or the programming of saccades. So here is our way to study face processing in the superior clicus. First, we train our monkey subjects to do a passive viewing task. So here is the monkey. The monkey needs to maintain fixation on a small fixation dot at the center of the screen. And we present object stimuli inside the superior clicus neurons receptive field. So what kind of visual stimuli we are going to use? Of course, there will be faces. And also to investigate how the neurons, whether they prefer face or not, we also include visual stimuli of non-faces, such as body, hand, fruit, and humanoid. But however, as I mentioned, the superior clicus neurons, they are strongly modulated by the visual salience. So how could we tell a neuron is responding to a certain object category like a face or body, or is just responding to a very salient visual stimuli? So here is our strategy. First, we include a lot of images in each object category, which is 30, and also they vary a lot in the visual salience. Some stimuli, they are very salient, and some stimuli, they are not that salient. And also, we carefully match the distribution of the visual salience across the 5 object categories by measuring the related low-level features, such as luminance, contrast, size, and also powers at 3 different spatial frequency bands. So using this kind of strategy, so we can directly test whether the SE neurons, they have some preference for certain object category that is largely independent of low-level features. So these are our just basic design of our experiments. So let's see what we find. So here we ask 3 different questions. So the first question is very straightforward, is do superior clicus neurons prefer faces? To directly answer these questions, we record 222 visually responsive neurons from the superficial and intermediate layers of the superior clicus. And we plot their average response to all the objects aligned on object onset. Here we can see the superior clicus neurons, they have an initial transient response to the object. And then there's a second lower peak. And then it just gradually decreases to a stable activation. And here is the responses to all the objects. And as I mentioned, we have 5 different object categories. So if we separate this response by the object categories, we can see there's already some modulation of the object category, and since there's some high response for faces even during the first peak, which is very fast. But we know that not all these neurons, they are object selecting. So to evaluate how many of the neurons they are modulated by the object category, we performed an ANOVA analysis with the factor of object category for each neuron. And here, this gray shaded area just tells you the proportion of neurons that are object category modulated, indicated by the significant ANOVA. Also, the time here is aligned on object onset. So here we can see during this early time period, about 50 milliseconds after object onset, there's already 50% of the neurons, they are modulated by the object category. Then we further ask, among these object selecting neurons, how many neurons they respond highest to face and to body or to other object category? And then you can clearly see this red curve, they just pop out. So this red curve just corresponds to the category of faces. So we can see during this early time period, about 50 neurons, 50% of the neurons, they are object selective, and almost all of them, they prefer faces. So this result here just tells you there's a prevalence of face preference for the SE neuron during this very early time period. Then we further ask, what is the magnitude of this face preference? So to answer this question, we just focus on all these early object selecting neurons. And then we plot their mean subtracted response to the 5 different object categories. So the mean subtracted response here is, for example, for face is first we calculate the response to face, and then we minus the average response across the 5 object category. And by this mean subtracted response, you can clearly see there's a relative high response for face compared to the other categories. But the interesting thing here is not only there's a preference for face, but how early it is. So if we select a window of 40 to 60 milliseconds after object onset, you can see this face preference is already very obvious. And as we know that the cortical face patches, the face-related response usually emerge about 80 to 90 milliseconds after object onset. This means that the face preference we observed here in the superior colliculus is even faster than the cortical face patches. And also the face preference we observe here is based on the average responses across these object selective neurons, and also by averaging the responses for the 30 different images for each object category. And then we further ask, how does this face preference look like in a neuron by neuron, and also in an image by image basis? So this heat map just tells you the results. So each row of this heat map just represents the results from one individual object selecting neuron. And also each column here just represents the neuronal response for one image. So there are 30 columns for face and also 30 columns for each of this non-face category. And for the color here, when it's greener, it's more green, it means response is higher. So we can see that this face preference is prevalent across these individual neurons. And also it's not caused by a very high response for a specific face image. It's caused by a high response for many face images across this face category. So based on all these findings, our answer to the first question is yes, we find the FC visually responsive neurons, they prefer faces with an extremely short latency that is even faster than the cortical face patches. And then we ask a second question is, how useful is this face-related activity in the FC? So imagine a downstream area that just fully rely on the inputs from FC. How well can it tell the current image is a face or is some other non-face image? So to direct answer these questions, we use the simple linear classifier analysis. And then these 4 different green curves just tells you the time course of the classification accuracy of 4 different binary decoders trying to classify face against each of the other non-face category. And then we can see here is aligned on object onset. So this classification accuracy becomes higher than chance level already very early after the object onset. And it already reached about 80% accurate about 50 milliseconds after object onset, which is pretty accurate in a very short time period. But however, the classification accuracy for the other decoders are not nearly as good as classification of faces. And here we also pick a time window of 30 to 70 milliseconds after object onset, and we construct the confusion matrix of the classification performance of all these classifiers. And for the heat map here, when the color is more red, it means it is more accurate. We can see during this extremely early time window, face is the only category that can be accurately classified. So based on these findings, our answer to the second question is yes, the presence of a face can be rapidly detected with high accuracies, just relying on the inputs from FC. And then we further ask the third question, is that given this extremely early face preference, what is the underlying neural circuit? So anatomically, the superior colliculus received direct input from the retina. It also received input from the visual cortex relayed by the LGN. And this direct retinal input to superior colliculus has been proposed to be a fast subcortical route for visual processing. Thus could be a very promising circuit for the face preference, the extremely early face preference we observed here. So if that is the case, if we can somehow block all the visual cortical input to SC, for example, by inactivating LGN, so if this retinal input to SC is really important for this face preference, so under this inactivation of LGN, it should not affect the face preference that we observed earlier. So to directly test this question, we record the SC neurons again, but now we start injecting mu signal into the LGN. And we want to evaluate how the inactivation of LGN affect SC's face preference. So during these experiments, the monkey subject is still doing the passive viewing task, but now we present the object stimuli inside the visual scotoma caused by LGN inactivation. The visual scotoma here is when we inject mu signal in LGN, and the certain areas of the visual field that the monkeys can no longer respond to the visual stimuli inside that visual field. And then this is measured by a separate visually guided scar task. And also at the same time, the stimuli we presented are faces and the two other non-face category, hands and human made. Let's see what we find. So first, before LGN activation, we again replicate our earlier findings that the SC display a short latency face preference. You can see here, even we include all the visually responsive neurons, no matter whether they're object selective, you can already see there's a high response for face. And for the ANOVA analysis, again, we find 50% of the neurons, they are modulated by object category at 15 milliseconds after object onset, and almost all of them, they prefer faces. So what happens if we inactivate LGN? Boom. We can see all the responses, including response to face and non-face stimuli, they are totally gone. And of course, for this ANOVA analysis, there's nothing there. And there are no neurons that are object selective, and there's no face preference anymore. So based on these findings, we find that the face preference in SC, they're actually not coming from the retina. They actually depends on signals routed through the visual cortex. So here is my summary. So first, we find the primate superior clicus display a short latency face preference that is even faster than cortical face patches. And then using classifier analysis, we find the presence of a face can be rapidly detected with eye accuracy, just relying on the SC responses. And using LGN experiments, we find the face preference and actually all the visually responses in SC, they depend on the visual cortex. So what is the significance about our finding? So first, I think because SC is one of the most important structure for orienting, for eye movements and visual attention, I think the SC here, they can just rapidly signal the presence of the face, whereas the later cortical face processing is slower, but maybe needed to recognize the identity of the face and a more dedicated face processing. And also our LGN experiments also emphasize the unexpected functional importance of the circuit from LGN to visual cortex to superior clicus during fast face processing. So finally, I would thank my collaborators in this paper. Leo Katz is actually the co-first author, Christian Kwaya and Adam Messenger, and my supervisor, Richard Crossley. And also many thanks for my lab members and my current collaborators. Thank you.